Agnieszka is director for Biokinetica, has been in the industry for more than 20 years and has covered more than 100 projects. I'm very happy to announce the topic for Agnieszka, and that is to cover the phase 1 unit's perspective. And after Agnieszka's session, we will have a discussion again, combining the last 3 sessions. So, Agnieszka, the microphone is yours.
Thank you. Thank you, Ronald. Hello, everyone, again. I hope you enjoyed your break. And I would like to thank you so much, Stephanie and Wendy, for presenting the sponsors and CRO perspective on the topic we are having now. And I will be extremely happy to present to you how Biokinetica managed the difficult time of pandemic as phase 1 unit. What major risks we identified and how we managed to mitigate them. Due to the time constraints, I will be focusing mainly on operational topics. However, I'm pretty sure that medical would be even more interesting one. I'm pretty sure that Perry will cover this in his presentation. I will try to provide you with some practical hints and practical aspects of what we have implemented at site to be able to continue clinical research during the pandemic time last year. This will be based on the case study that Stephanie and Wendy already described. So the healthy volunteer phase 1 study with a biosimilar compound. However, as you can imagine, it's really difficult to separate the experience from one study to another. So I believe that our experience I will be sharing with you can be applied to any study that we've been conducting since the pandemic hit us last year. And I also believe that this could be also well utilized in any clinical research, regardless of its phase or geographical location. So I hope you can see my slide. Just briefly about who we are. Biokinetica Clinical Research Site is a dedicated phase 1 unit with 30 plus beds. We can accommodate up to 50 subjects at the same time. Of course, during the pandemic, we imposed some restrictions on that. So currently we are able to accommodate 30 subjects, 24/7 medical monitoring, internal in-house emergency care unit with 5 beds. On the same level, we have direct access to cardiac emergency care unit of the cardiology hospital, which is on the same floor in the same building. In-house pharmacy area, in-house laboratory for processing of blood and urine. At the same time, we do have our own in-house operational theater. And last but not least, and this is of course related to our today's topic, we have our in-house Pcr facility. This is rapid PCR, and this allows us to perform a large amount of testings, pre-screening, and during the study on ad hoc basis, if needed, also in our personnel. So the results we have is within 20 minutes. Of course, we do have the collaboration with the full PCR accredited laboratory we are using within the clinical research protocols. Okay. Some pictures showing the COVID premises. So as you can see, it's divided into the isolation area. We have ambulatory part for taking the swabs, as well as the laboratory itself for this rapid test, equipped with laminar, chamber flow, etc. Okay, so those who know the Polish situation last year knows that the first wave was coming quite slowly, but the situation was that fall last year when we were facing the second wave of the pandemic, Biokinetica was at the stage of the startup of several large healthy volunteer large cohort studies. Not only biosimilar studies, but also real phase 1 studies with first administration of IMP to healthy volunteers. So we were in a bit uncomfortable situation. We found ourselves, however, we had some time to prepare, of course, from the first wave. So we, together with and across all departments, with our quality department, laboratory department, pharmacy department, last but not least, medical department, we sat together and, of course, discuss all potential risks. And we tried, of course, to find answers how to mitigate them. What we knew then, and probably other words, what we didn't know, because honestly speaking, there were more question marks than answers then. So of course we had to define the operational risks and the medical at the same time. As I said, I will be focusing on operational topics today, and how to manage the new situation we were in. Within the absolutely unknown regulatory landscape and ethical requirements, because at that time we didn't know what and how the regulations would change, how the Biotics Committee, our local Biotics Committee regulation will change towards different protocols we have, biosimilar with healthy, phase 1 with oncology patients, real first-in-human studies with different compounds having different immunogenicity, etc. How this would impact our recruitment, and how to deal with that. Of course, we needed to discuss internally whether any organizational changes will need to be implemented in terms of the personnel, shifts, infrastructure. So this was one of the key aspects that we needed to discuss. Then coming to the communication with the sponsor, with the CROs. Some of them, of course, put their studies on hold, were considering changes into the study protocols. Some of the studies were put on hold and did not start since then. We were asked what you think about the Biotics Committee requirements, how we should update or include some additional procedures into the protocol. So this was, I would say, a listing of question marks we had to answer together with our sponsors and CROs in, of course, very open communication. What our conclusions were at that time, first of all, very quickly, we have identified that we need to implement our in-house internal COVID-19 risk assessment and risk mitigation plan, because we had no state rules, Biotics Committee requirements clear enough to navigate us through the new situation. And of course, this was really important. This was already stressed out by Stephanie and by Wendy. Clear and very open communication with all stakeholders. So with the team, with the subjects, with sponsors, with CROs, with our vendors, with our landlord. So with everyone, so that, because we have to do it together. Yeah, we cannot act alone. Okay, then. So first, having in mind that we were just about to start a large cohort bioequivalence, biosimilar studies. We had to focus on several aspects to assure the smooth conduct of those studies, meeting the timelines, meeting the recruitment, of course, deadlines and recruitment goals that we discussed with the sponsor and with the CRO. So those, I would say, measures were split into two, let's call them sections. So first of all, our in-house procedures and instructions that were not related directly to any of the protocols. So very quickly we have drafted and finalized, and it's still a working document that now I believe it's sixth version, the COVID-19 risk assessment and risk mitigation plan. Second, we have openly discussed the protocol elements that we need to include, and we have also discussed them with the bioethics committee, and they were also discussed during the bioethics committee meeting by our investigators. Then moving to the organizational parts, how to organize the cohorts, how to split the cohorts at the screening stage, at the in-house stage. We have to remember that those biosimilar projects are large cohort projects. So we could potentially treat even 20 up to 30 subjects on one day. Of course, we had to change it. We had to first split the groups into smaller cohorts, but this was not only related to the epidemic situation, but also to the fact that the recruitment rates, what Stephanie and Wendy mentioned already, were lower. We could not have like overlapping groups of in-house subject, of screening subjects. So we had to organize dedicated separate premises for the screening activities. Then moving to the organization of the team's work, duties. We have to remember that phase 1 trials are very requiring, I would say, plenty of personnel being at the same time in the clinic. Dozens of nurses, several investigators, laboratory technicians, pharmacists. So we had to assure that we are working in a separate double teams. And this is how this was organized, so that in case of any problem, any infection among the personnel, we can continue the study if we have no, of course, major collapse because of the infection, and we hadn't, fortunately. Adaptation of the clinic, I already mentioned, but not only the clinic itself, but also building up the dedicated COVID-19 facility, providing, consisting of the isolation area, because we have to remember that in phase 1, real phase 1 trials, when we, of course, dose the subject, we have to monitor them. We have to monitor them sometimes until 2 weeks' time, and we should be able to monitor the parameters, medical parameters, even if they have some medical and COVID-19 symptoms. So this area was dedicated for such situation. PCR testing, this is really a challenging topic, because of course we had to find a vendor for full PCR testing quick enough to provide us with the results within maximum 24 hours. This was hard, as you can imagine, because of the situation in Poland. So new, of course, laboratories were developed, but still we wanted to find a reliable vendor that would provide us the result as quickly as possible, at the same time building up our infrastructure for rapid COVID testing. But this was from the beginning dedicated for pre-screening testing of subjects according to our internal procedure. Then moving to regulatory challenges, I have already covered this a little bit. So as we have already discussed with Ingrid, there were no clear regulations from the beginning, either from the regulatory body or bioethics, local bioethics committee. I know from my Dutch colleagues from ICON side that they had some specific guidelines from their bioethics committees for phase 1 trials. We didn't have. So as I said, we had to act proactively and to develop our own instructions, procedures, to implement them, and of course to update them on regular basis, looking at the situation. Looking back, I would say that each study was treated a little bit different by the bioethics committee. Of course, it always depends on the reviewer, but in most of our biosimilar studies, and the one that we are discussing today as case study was the case, the first prolonged hospitalization period was implemented and required by the bioethics committee, and second, the quarantine, so post-admission quarantine. In the case of this study, this was the home quarantine. As Stephanie already mentioned, we had to contact subjects on daily basis to confirm that they are in quarantine. We, of course, kept the records for that. We had trained personnel doing this task. In other clinical trials, very similar one, the hotel quarantine was implemented into the protocol. So we had, and we did, find a vendor for that, so a nearby hotel in which we could assure a separate safety place with single rooms, so that subjects were put in quarantine before admission to the site, after the COVID testing, and then after the discharge. Of course, very good collaboration, very good communication with all stakeholders, with our bioethics committee, local bioethics committee, having at the same time open discussion with sponsors and CROs were also necessary to navigate, to manage the regulatory and ethical risks at that time. Recruitment challenges. This is something that is really interesting because, as Wendy said, it depends, and I can imagine it depends from the site to the site. At first, we thought, and we have discussed this with our recruitment team and top management, that probably the new situation would put us in a position that we would have more healthy volunteers willing to participate in the studies because simply of them getting unemployed. This was partially the true, but very quickly we have realized that we have to recruit much more subjects, healthy volunteers, to be able to fulfill the recruitment goals. This was because of the fact that, as Ingrid already mentioned, this was some kind of general fear in the society, and subjects simply did not appear on the pre-screening COVID testing. So normally our dropout ratio at the screening is like 15 to 20%, and we face the situation when 50% of subjects invited for pre-screening COVID did not appear for the visits. So, of course, very quickly we have to adapt. We were discussing this with our recruitment team. We had to change our recruitment plans, recruitment goals. And of course, that is why we also invented and implemented a wide recruitment campaign, simply because to, of course, find a new pool of patients, new pool of subjects, healthy volunteers to our database. But recruitment was not also the only challenge in terms of contacting the subjects. Of course, we had to implement additional measures and activities targeted at contacting the subject on a regular basis, almost on daily basis. So even after the discharge, during the ambulatory part of the studies, dedicated personnel, our call center, call subjects, confirm their status, health status, if any COVID symptoms occurred, etc., so that, to be able to manage the potential dropouts, potential not compliance in subjects appearing at the following ambulatory visits. We were quite surprised, however, with at that time with the low positive PCR ratio. So I have some statistics here. So almost out of almost 1,500 pre-screening tests, which we performed between December 2020 and February this year, we had less than 50 cases. So this is around 3.5%. So at that time, knowing the, let's say, general statistics of the positive tests performed, this was quite surprising. And what is very good information, until today, we have no positive COVID subjects during the in-house period at site. But of course, we have the instruction and the risk mitigation measures, and we are ready for such a situation. I'm pretty sure this will be covered somehow by Tomasz, but just to give you the information about the data entry challenges that we faced in clinical trials, phase 1 clinical trials. As you know, the timelines in those trials are very tight. So generally, the expected data entry time is like 24 hours, 48 hours, sometimes 72 hours. And of course, this was the first thing that we had to openly discuss with our CROs and our sponsors. We are focusing on clinical operations. We are focusing on recruitment to meet your timelines, your goals, our joint goals. But we have to negotiate in terms of data entry timelines. And I must say that all our CROs, our sponsors, understood the situation. We're very open to discuss it in an openly manner. We have prepared updated data entry trackers that were followed by the team. Of course, we had to recruit additional personnel, data entry personnel, to be able to continue and to meet the deadlines in case of any positive colleague in the team. Okay. And I believe this is last but not least the budget challenges. Some of them were directly related to, of course, to the protocols and to the new procedures that have been implemented into the protocols and were, of course, covered by the sponsors and CROs. Some of them, on the other hand, were something that allowed us to continue on our operations, were understood, of course, by the sponsors and CROs, but were not directly, of course, included into the protocol. So, yeah, this is just to list some of them. We had to invest in additional dedicated infrastructure, COVID-19 premises, I already mentioned. Learning curve costs. So, of course, from the very beginning, everything was new for us. We have adapted to the new situation. But only after, let's say, several weeks, several months, we were in a position where we are fully operationally capable of continuing our studies. Additional recruitment efforts, I already mentioned. Recruitment and training of new personnel. But also, and this is something that I believe needs to be stated here, at least this was the case in Poland, we had to somehow change the employment structure of our personnel, especially medical personnel, because of course, nurses, investigators working on daily basis in large public hospitals were not so allowed to come to different places. So we had to change our employment structure to permanent contracts with most of our nurses and our investigators. Change organization of visits, so smaller groups of patients, smaller group of subjects. This is also, of course, and this also has impact on the budget. Change shift structure. So smaller number of people in the team, but more teams, overlapping teams, of course, assuring the continuity of the clinical research, clinical activities of our projects. As for the conclusions, so first of all, and again, I would emphasize that phase 1 trials are connected with a very high operational risk and medical risk, of course. And this is regardless of the fact that we are during the pandemic. And during the COVID-19 outbreak we are facing for the last year and a half, this put us in a position that we had to change our mindset. We have to think out of the box. We have to be proactive. We have to do daily assessment of the situation. So as Stephanie mentioned, one day you close your computer, another day it seems that probably some of your studies needs to be put on hold, or your planned efforts, activities, clinical activities needs to be changed because of new governmental restrictions. But what is important here and what helped us to overcome those obstacles? It's necessary for all stakeholders to communicate in an open manner, in a daily manner, to inform our sponsors and CROs about any additional restrictions, problems, difficulties we may face, we are facing, or we may face in the coming days. But also internally, to discuss on daily basis across the teams, with laboratory team, with technical team, with medical team, with pharmacists, with recruitment personnel, on where we are, what's the status of the project, how the new information that were coming could have an impact on their operations. But I would say, and I believe this is the most important thing that we have learned, the idea that is very, I would say, constructive and optimistic, that these challenges that we faced were at the same time a driving force for clinical sites as Biokinetica, and stimulated us to do changes, to implement new measures, to be able to continue our clinical research. And this thankfully allowed us to continue undisturbed clinical trials in line with the scheduled timelines, with the deadlines agreed with the sponsor. And of course, at the same time, ensuring the safety of our subjects. So thank you very much. I will be very, very happy to answer your questions. Please feel free to put them in the chat. And the very end of my presentation, I would like to share with you a very nice phrase. I'm not sure who wrote it, but it's like this: sometimes you have to feel rain under back of your neck. Getting uncomfortable makes you realize what you need to change. And this is what happened with our clinical research unit. And I hope that you will enjoy further discussions and further seminar presentations. Thank you very much.
Thank you, Agnieszka. I think indeed we were all a bit uncomfortable over the past 2 years, to put it like that. But I think you've done an excellent job in keeping the unit operational and safe for all the subjects.